Okay, all right. So today is March 23rd, 2021. It's the third day of our 7th unit. We're going to continue with the discussion we began yesterday about inheritance. So please make sure you have access to your notes because we will be following that same mechanism. I want you all to have access to the steps of that process. Today, we're going to deepen our knowledge about inheritance and discuss 2 additional inheritance patterns. One of them is called codominance, and the other is called incomplete dominance. But our essential question remains the same. We're just continuing on with the same conversation. All right. Yesterday, we kind of went through what am I trying to say? Practice of cold calling, which is something that many of you will probably experience in college. And if you go into grad school or law school or med school, you'll be in these lecture settings or these seminar settings, and your teachers will call on you without warning, and you'll be expected to give the answer for the whole class. I want to continue what we were doing yesterday. It actually worked really well. I think you all were pretty engaged and you were following along really well. But today I want to engage the folks at home a little bit more. So please, if you hear your name called, even if you are attending class virtually today, please make sure that you're ready to go and give us a good answer. All right. We're going to start off with some review of the warm-up. I believe this was one of the warm-up questions. Hopefully it was. I'm pretty sure I saw this in the warm-up. The question was, a strand of DNA reads AAC, CGG, GCA. What would be the amino acid sequence produced from this DNA? So before we do that, before we can actually figure out the amino acid sequence, we've got to find out the mRNA that is copied from that DNA. What process, what's the name of the process? Who do I want to hear from? Janelle, you're first. You're the first name I see. What's the name of the process that takes us from DNA to mRNA? What's that called? Janelle? Or, oh, there it is. Thank you, JT. That's transcription. Yeah, to go from DNA to mRNA, it's called transcription. So we've got to do that first before we can actually go on to amino acids. Sally, what do we transcribe A to? A is, and which letter of mRNA? A is transcribed to what? U, excellent. So the first and second bases are both A's. They both get transcribed to a U. Ashanti, what about a C? What does a C get transcribed to? G, good. So the next 2 bases become a G. And then Araceli, G gets transcribed to what? C, good. So the next 3 bases become a C. We already had Ashanti tell us that C is transcribed to G. And then Sally told us that A is transcribed to U. Now, I tried to trick you all here because I did not include any T's in the DNA. So some of you were probably tempted to go on and translate that strand of DNA immediately because you don't see any T's there. But as JT tells us, we've got to transcribe DNA to mRNA first. All right, now that we have these mRNA, and they're grouped in a specific way, they're grouped in groups of 3. What are those called? Does anybody remember? When we group them in groups of 3? Excellent. Thank you, Sally. Yeah, so when we group them in groups of 3 letters like UUG or GCC or CGU, we call those codons. Now we've got them grouped. We can actually look at this codon chart to figure out the amino acid. Let's see. JT, since you gave us transcription, what's the process called to go from mRNA to amino acids? Yeah, the name of the process. Going from it. What'd you say? No, not the name of the actual amino acid, even though that's correct, but just the name of the process to go from mRNA to amino acids. Translation, excellent. And JT did go ahead and give us the correct amino acid, but I'm going to ask, let's see, someone at home, if they can also give us that very first amino acid. UUG, what does it become? Someone at home. Which amino acid does that give us? Thank you, Melissa. Yes, that does give us leucine. The abbreviation is LEU. Now, if you're confused, which would be okay, it's been a few weeks since we first introduced this, then we need to use that codon chart that's on the left side of your screen. There's also a codon chart that is bookmarked, or it's available to you all in the Unit 5 modules, and I encourage you to bookmark it. But with this first codon, we've got UUG. What we need to do is start off in the middle of that circle, and we're going to move outwards, kind of like a ray of sun. So we start off in the middle. The very first letter of this codon is U. So now we're only looking in the very top right quadrant of that image. We're only looking in the section that gives us U, that's in the top right. The second letter is also U. So now we're only looking in the section of this codon chart that corresponds with UU. We're moving outwards. And the last letter is G. So we're only looking in the section that is G. For my folks at home, I'll finger it on the board. So up here is G. We're only looking in this section. And then the second letter is I'm looking up here. Only leaves me with two options. It could have been phenylalanine or it could have been leucine. But then that third letter is G. So I'm moving outwards. G is my third letter. So that tells me JT was right. It's leucine. Okay. Tayshawn, do you think you can give us the second codon? Oops, I almost showed it. GCC. Here. And since Tay is not in the chat, then somebody at home can definitely jump in with the answer for the second codon too. What did you say? Oh, okay. No. Okay, so what's the very first letter of this codon? You too, Abigail. You're gonna try again. With the,
Is it Allison? It's like ALANI,
yes,
something like that.
yes, that's good, Lisa. Thank you. That's correct. Did you hear that, Jay? Okay, Tayshawn. What's the first letter that we're looking at in this codon? G. Yeah. Because this is the very. So that means we're only looking in the top left portion. We're only looking up here, right? And the second letter is what? C. Good. So we're moving outwards now. We started off here, then move outwards. So now we already know. We don't even have to look at the third letter. Because GC, that only gives us one option. Alanine. Okay, so this one is alanine. Good, good, good. Okay, and then Abina, I told you I was coming to you last. Do you have the answer for me? What is it? You can just give me the abbreviation. Good. ARG. Excellent. This is arginine. Arginine. Good. So we can't get away from this. I can almost guarantee there will be at least one question on your EOC exam about this. We'll keep coming back to it. I just don't want you all to forget how to use these codon charts. Good job. Okay, yesterday we defined complete dominance. You should have already written this down for those of you who were here yesterday, so nothing new here. But just briefly, we talked about complete dominance. It's cool because you either have the trait or you don't. As we define two additional types of inheritance today, the thing that we need to keep in mind is the heterozygote. There will be differences in the heterozygote. The dominant and the recessive, those don't really change. It's the heterozygous genotype that's going to be kind of funky as we define these next two. So with complete dominance, the heterozygous genotype shows the dominant trait. Okay, we're going to see that that's not exactly the case over the next couple of types of inheritance. Not all traits are as simple as complete dominance. In fact, we would be a very black and white world, a very cut and dry world if everything followed complete dominance. We would only have one of two types of things. In reality, we know not only are there some traits that are defined by multiple genes, but there are also some traits that result in what we would consider blended phenotypes, and then also some that result in spots and stripes or phenotypes that are being displayed at the same time. So let's take a look at these. Here's a new one that we're discussing today. Incomplete dominance. In this type of inheritance, the dominant allele does not completely mask the recessive allele. So the heterozygote shows a blend of both. You guys can, I would definitely recommend drawing this chart too. It's not going to be on a slide, but it could be helpful. So my people at home, I'm drawing a chart that I think will be helpful to you all to better understand some of the differences between these types of inheritance. Okay, so I'll move out of the way eventually, but let's move on. So we've got a few examples down there at the bottom of the screen, and these are just for human traits. We see this happening. We see incomplete dominance a lot when it comes to other animal species as well as plant species. But the other thing to note here is that the notation is slightly different. So in complete dominance, we use a capital letter and a lowercase letter. With incomplete dominance, we use capital letters the entire time, but in order to notate the recessive allele, we use an apostrophe. And we call that apostrophe prime. So R apostrophe would be called R prime. And that represents the recessive allele. Any questions about that? No? Okay, we're good. Okay, so here's an example in flowers, pretty common. Here they actually do use a lowercase a. Ignore that. We like to, in North Carolina state standards, you will see an apostrophe. So 2 capital R's, that's the homozygous dominant genotype, and it gives you a red phenotype. R prime would be the homozygous recessive genotype, and it gives you the phenotype of white. So we say that red is incompletely dominant to white. So the heterozygote, because this is incomplete dominance, is going to show a blend of those 2 traits. And we call that, again, the heterozygous genotype would be R prime. And it shows the pink phenotype. So it's a blend, of course, red and white mixed to make pink. Okay. So we're still following these same steps to solve a Punnett square. Nothing changes about that. We just now have this kind of new rule that we're going to talk about. We are going to do some examples. Well, just one example with incomplete dominance, but I liked how it worked yesterday where I kind of just gave you guys a timer and I walked around and was checking your answers. So we'll do that again. And you'll have a lot more time to work independently today once we get through these examples. So, of course, you determine what each allele means and you make a key. You're going to split the parental genotypes on the sides of the box, on the outside of the box. You're going to combine them together, and that is what we call a cross. And then you'll analyze to determine the genotypic and phenotypic ratios. And of course, answer whatever question is being asked. All right. So there is an assignment in Canvas called U7D3 Whiteboard Practice that I would like you all to just open up. These are going to be example problems that you'll be able to work through in today's class period. We're going to do the first one together. Does anybody not have a Chromebook? No, everybody's got a Chromebook. And then I'm also going to ask you all to turn this in on Canvas once it's done. So this is worth points. We're just going to do the first one of each type of inheritance together. Okay, so once you open it up, please make sure you change the name of the document, of course. Make sure that your name is in the name of the document. And then also don't forget to save it to your biology folder. I'll wait about 30 more seconds. And I'll step out of the way so you guys can see this. Okay, so let's do this first one. Mine is going to look slightly different than yours, but it's the same question. So it says brown is incompletely dominant to white in mice, resulting in a tan phenotype. If a brown mouse with the genotype BB mates with a white mouse with the genotype B prime B prime, what phenotype would their offspring have? All right, so I think we're in a good place to answer this question. What's the first step that we always follow? Make a key. Excellent. Our key still looks exactly the same. We still need to know the same information. What is the dominant allele? What is the recessive allele? And what do each of the three genotypes look like? And what phenotypes do they correspond to? But nothing changes there. So, Araceli, what's the dominant allele in this case? It's just a B, a capital B. And which trait does that correspond to? Brown. Excellent. And then who else have I haven't called on you, Deasia. What's the recessive allele here? What'd you say? I can't hear you. Okay, so we still are using the letter B. Let's look. So if we're looking here, the white mouse represents the recessive. So in this case, the recessive allele would be with an apostrophe. And that B prime gives us which trait, Tay? White. Good. Okay. All right. And Sally, what would be the homozygous dominant genotype? Capital B, capital B. And which phenotype is associated with that? Brown. Good. Abina, what is the homozygous recessive trait? Say it one more time. Not the heterozygous. The homozygous recessive. This B prime. It'll still be capital, and they're both B prime, B prime. So homozygous, of course, just as a brief reminder, I know you guys know this, but homozygous means same. So because it's homozygous recessive, we need the recessive allele and we need it twice. So B prime, B prime. And Abina, which phenotype does that give us? White, good. And JT, the heterozygous genotype, what is that? B, B prime. And here's the tricky one. Which phenotype does that give us? That gives us tan. So this is a little bit different. Can you guys tell that that's tan? I guess so. This is a little bit different than what we've done in the past, right? Because in complete dominance, the heterozygote gives us the dominant trait. But with incomplete dominance, the heterozygote gives us a blend of the dominant and recessive traits. So brown and white blended gives you tan. So that's the difference here. That's what we mean with incomplete dominance. But good job. Now that we've completed the key successfully, we are in a great place to go forward. All right, now I want some information from some people at home. What's the next step after we've made the key? What do we do after that? Anybody at home? Hello? What do we do after we make the key?
Make the Punnett square.
Yeah, we make the Punnett square. We need to, as Melissa says, split the parental genotypes. So in this case, we're given the parental genotypes. It would have been a little more tricky had we just said brown mouse and white mouse, but we're actually told BB and B prime, B prime. So that makes it a little bit easier for us. So parent one is the brown mouse. We put those two alleles on top of the Punnett square. We split them up. So one B goes over column one, the second B goes over column two. Then we split up parent two's genotype, B prime, B prime. One B prime goes to the left of row one, and one B prime goes to the left of row two. And now we're actually going to do the cross. So yes, B prime. Yep, B prime. And the Asia, what's going to go in this box? Hmm? Excellent, yeah. Good. So in this case, all four boxes end up looking exactly the same. All four boxes have the heterozygote. So which phenotype is that? The question is asking what phenotype would their offspring have? Tan, good. And this is where it's important that you have the correct key. Making your key is so, so important. Okay, because once we see the genotypes in the Punnett square, if we've got a good key, we already know what phenotype that is. Okay. All right. So let's pause here, and I'm going to give you guys some time to fill out that very first question in the U7D3 whiteboard practice. Let's take 2 minutes to fill that out, and then we'll move on. Yes, sir. I'm doing well. Barnes. Yes. Deasia. Yes, you may. Thank you. Okay, about 30 seconds left, folks. 30 seconds. All right, so let's move on. We'll talk about the next one, the next type of inheritance now. Okay, so this third type of inheritance that we'll discuss is called codominance. So you've got complete dominance, incomplete dominance, and now codominance. In this type of inheritance, the two alleles are equally dominant. And the heterozygote in this case will show both phenotypes simultaneously. So this could look like spots or patches. And in some cases of livestock, you might hear the term roan being thrown around. The notation in this case is that we use two different letters. They're both capitals, but we use two different letters. So one capital letter represents the dominant allele. Another capital letter will represent the recessive allele. Any questions about codominance right now? Okay. Okay, so one more example, and this is taken from U7D3 whiteboard practice. You'll have to scroll down to the codominance section, but it is there. The question says, if black chickens with the genotype BB are codominant to white chickens with the genotype WW, show a cross between a black chicken and a black and white chicken. So the procedure does not change. The very first thing we need to do is, of course, make a key. And the key does not change, looks exactly the same. We're looking for the same information. We need to know the dominant allele. We need to know the recessive allele. We need to know what the genotypes look like and which phenotypes they correspond to. So, Abina, let's just start off with you. What's the dominant allele in this case? B, and which trait does that give you? Black. Sully, what's the recessive allele? W, and which trait does that give you? White. In this case, I did use the word recessive, but in reality, it's not really recessive. Both of them are equally dominant, okay? But that's what I was looking for. So thank you, Abina and Sally. Tayshawn, what is the homozygous dominant genotype? B. Remember, we always need two. BB, good. And which phenotype does that correspond to? Black, good. Araceli, what's the homozygous recessive genotype? Two W's, good. And which phenotype does that correspond to? White. All right, JT, somehow you always end up with this harder one. What's the heterozygous genotype? BW. And which phenotype does that correspond to? Both black and white. Good. So again, in the case of codominance, we see both traits appearing in the heterozygote. You see both of them happening simultaneously. Okay. After that, we're going to start making, as Melissa put it earlier, we're going to split the parental genotypes. So again, in this case, we're given the parental genotypes. I imagine that in some of the practice problems you all will do, that will be kind of the source of difficulty, is figuring out what the genotypes of the parents are. So please make sure that you're careful when you're doing that. Take your time, really think through what the question is giving you so you can figure out the correct parental genotypes. If you don't figure out the correct parental genotypes, it's not possible for you to get the right answer. Okay. So in this case, we've got one black chicken that's BB, and then we've got one black and white chicken with the genotype BW. So Ashanti, what is going to go in the top left box of our Punnett square? Capital BB. Good. And Deandra, what's going to go in the top right box here? Capital BB. Good. And being I'm swinging back around to you, what's going to go in the bottom left box? BW, excellent. And then Sally, just finish us off, please. BW, good. So it seems like you guys have got the Punnett squares pretty solidly down. In this case, it didn't exactly ask a question, but what information can we figure out from our results here? What do we know? Just throw something at me. Now, be careful, because in this case, it's not a blend. So, Sally said 2 black, which is correct. We've got 2 BB, and yeah, we got 2 BB. And then she said 2 gray. I'm glad she made that mistake because it can get confusing between incomplete dominance and codominance. Incomplete dominance would be gray. You would have been right with it if this were incomplete dominance. But this is complete dominance. So we see both black and white appearing at the same time. There are, you can actually look up a picture of this on Google. There are some chickens that have both black and white feathers. So that's good information that we can take from it. That's the phenotypic ratio. What about the genotypic ratio? What else could we say about it? What percentage of the chickens are heterozygotes? 50% of the chickens are heterozygotes. What percentage of the chickens are homozygous recessive? Use your key. Homozygous recessive. 0%. Excellent. Good. And then, of course, 50% of them have to be homozygous dominant in that case. All right. So we've got 22 minutes. That took longer than I thought. I want to see you guys work on the whiteboard practice. There is another assignment there called incomplete and codominance inheritance practice. I'd rather see you work on the whiteboard practice. That way I can kind of walk around and check. And I'll also be looking at Dino for those of you who are at home. And that'll be good practice for us today. So we've got 22 minutes to make progress on that. But it seems like you guys are grasping this pretty well. So in here. For those of you who are at home, please do make sure that you jump back into that assignment to use your time wisely. And I'll go back to this one so you guys can see. What's 7 times 7? Thank you. or would like me to check their problems? Okay, folks, thank you all. Excellent job today. It's been a strong week. We'll discuss tomorrow. We do meet earlier tomorrow because of it's a virtual day, so I think we meet at, anybody know off the top of their head? 10 o'clock? Yeah, thanks, Ted. 10 o'clock, so 30-minute class tomorrow. It'll be quick. Talk to you guys then.